Have you ever pondered how minor genetic alterations can lead to major health implications? Today we're focusing on chromosomal translocation, a common genetic abnormality characterized by the exchange of chromosomal segments between non-homologous chromosomes. Think of it as a puzzle where pieces from different puzzles are forced together, leading to a distorted image. In the genetic world, this distortion can lead to diseases, most notably cancer. But how does this happen? It starts with DNA double-strand breaks, moving on to repair processes like non-homologous end joining and homologous recombination, and culminates with the involvement of specific genetic elements. This complex process is not just a random occurrence, but a meticulously orchestrated event that, when goes awry, can lead to severe health implications. Understanding this process is our key to unlocking new ways of treating translocation-associated diseases. So, let's journey back to the beginning, to the discovery of chromosomal translocation. The discovery of chromosomal translocation dates back to the early 20th century. At that time, scientists were just beginning to unravel the complex world of genetics. A key breakthrough came when they observed the peculiar phenomenon of chromosomal segments moving between non-homologous chromosomes. This was a revelation that challenged the established understanding of genetic inheritance. Pioneering researchers like Barbara McClintock were instrumental in these early days. McClintock's work on maize plants led to the recognition of this remarkable genetic event. She observed that genes could move and rearrange themselves on a chromosome, a discovery that won her a Nobel Prize and laid the groundwork for our understanding of chromosomal translocation. These early observations marked a turning point in the field of genetics, opening up new avenues for exploration and setting the stage for decades of research. As research progressed, scientists started to unravel the mechanisms leading to these translocations. The 21st century brought with it an era of advanced genomic technologies and molecular techniques. These advancements have been instrumental in understanding the mechanisms of chromosomal translocation, a prevalent genetic anomaly that plays a significant role in various diseases, including cancer. One of the key mechanisms that drive chromosomal translocation is DNA double-strand breaks, or DSBs. These breaks can be induced by both internal factors such as reactive oxygen species and external agents like ionizing radiation. When DSBs occur at different locations on the chromosomes, the repair process can sometimes go awry, leading to the fusion of broken ends from non-homologous chromosomes. This fusion results in translocation junctions, essentially swapping segments of DNA between two chromosomes that typically wouldn't interact. The repair processes that rectify these double-strand breaks are critical players in the formation of chromosomal translocations. Two of these processes, non-homologous end joining, or NHEJ, and homologous recombination, or HR, are particularly noteworthy. NHEJ is a quick and dirty repair process that often leads to errors, while HR is a more accurate but complex process that requires a template for repair. When the regulation of these processes is disrupted, it can lead to genomic instability and disease. Additionally, specific genetic elements can also influence the formation of translocations. For example, certain sequences of DNA can be more prone to breaking and rejoining, thereby increasing the likelihood of translocation events. Understanding these mechanisms is not just an academic exercise, it holds profound implications for how we diagnose and treat diseases associated with chromosomal translocations. Unraveling the intricacies of how translocations occur can provide valuable insights into disease development and progression, and potentially lead to novel therapeutic strategies. With the understanding of how translocations occur, it was time to delve into the consequences of these genetic events. Chromosomal translocations, while microscopic, can have macroscopic effects on genome stability and function. These rearrangements can trigger an array of impacts that disrupt the harmonious operation of our cells. One of the most significant effects of translocations is their ability to meddle with gene regulation. They can lead to the formation of what are known as fusion genes. Picture two different genes coming together to form a completely unique entity. This new fusion gene can potentially go rogue, acting independently of the body's normal checks and balances. In the context of cancer, this can result in the uncontrollable growth and proliferation of cells, driving disease progression. Translocations don't stop there. They can also interfere with normal cellular processes. Imagine a finely tuned symphony being abruptly interrupted by an off-tune instrument. This is what translocations do to our cells. They disrupt the normal rhythm, contributing to the development of disease phenotypes and even resistance to therapies. The cancer context again serves as a prime example. Translocations can lead to the dysregulation of oncogenes or tumor suppressor genes. These are genes that, when functioning normally, either promote cell growth or halt it. But when translocations disrupt their regulation, it can set the stage for tumorigenesis and tumor progression. The profound consequences of chromosomal translocations have led scientists to explore potential therapeutic interventions. The hope is that by understanding these translocations better, we can develop ways to either prevent or correct them, ultimately improving patient outcomes. Understanding the intricate dance of chromosomal translocation has opened new avenues for therapeutic interventions. As we delve deeper into the molecular mechanisms that underpin these translocations, we uncover potential strategies that could disrupt these events or mitigate their downstream effects. Consider DNA double-strand breaks, or DSBs, a key player in translocation formation. If we could find a way to prevent the misrepair of these breaks, we could potentially interrupt the formation of harmful translocation junctions. This is but one of the many possibilities that our increasing understanding of translocations presents. Furthermore, the discovery of translocation-mediated fusion genes, which can dysregulate oncogenes or tumor suppressor genes, provides another promising target for intervention. By disrupting these fusion genes, we could potentially halt the progression of certain cancers at their source. But it's not just about prevention and disruption. Advances in genome editing technologies such as CRISPR-Cas9 have given us the tools to correct existing translocations and restore genomic integrity. This could pave the way for more effective treatments for translocation-associated diseases, offering hope to those affected. As we continue to deepen our understanding of chromosomal translocations, we are continually presented with new opportunities for therapy and intervention. Continued efforts in understanding the complexities of chromosomal translocation promise a future of novel therapeutic strategies targeting translocation-associated diseases.